things look different from those things, no question. Horses do weird stuff where it's like basically one big toe, right? That's their hoof. Horse legs, one bone, two bones, little blobs, hoof. Same thing all the time. Now, your options are pretty limited, and God likes it that way. You know, that doesn't get it done. It's true, but it doesn't answer the question we're asking. What's your only other option besides common ancestry here is to say there are design constraints that force this choice. It's the only good way to do it. We await findings to confirm that hypothesis. Perhaps you might think of it as a research project. My advice is you shouldn't waste your time, but it's the only other sort of living hypothesis. One bone, two bones, little blobs, digits, everywhere on the planet. Oops, I'm not there yet. We got a couple other examples of development in case you don't think tetrapod limbs are that interesting. Did you know that you can make chicken grow teeth? Yeah, so chickens. Is that a hint? Chickens are birds. Birds are thought to have descended from dinosaurs. You probably have heard that one before. It was somehow the centerpiece of Jurassic Park and warm skin and quick versus slow and yada, yada, yada. But the point is, birds are thought to have descended from dinosaurs, and dinosaurs are reptiles. And so birds and reptiles are thought to be, fairly closely related, as weird as that sounds. Well, every now and then, there's a mutant bird that turns up that has teeth. Now, first of all, that tells you that birds can grow teeth, but don't usually. But second of all, those teeth look interesting. They look a lot alike alligator teeth. They're a certain kind of tooth. Well, a group studying this, they found a mutant chicken. By the way, how you find a mutant chicken that has teeth, I'm not quite sure. They were scientists. They weren't, egg layer, checking the mouths or anything like that. But anyway, these guys found a mutant chicken that would often grow teeth, and they went to study what happens, and they found a particular signaling pathway that's messed up. Biologists in the crowd, beta-catenin is all you have to overexpress in that tissue to grow teeth. And if you know what beta-catenin is, that should surprise you. It's a pretty basic signaling molecule. Overexpress it in that tissue, whoop, they grow teeth. Teeth, one protein. Now, why is that interesting? Well, because the bird's using the same tools to build its body as the alligator is. They got all these tools from their common ancestor. What the bird did was turn off some of those, say, dude, the teeth don't really help anymore. It's pretty easy to turn them back on. Why? Good design? It doesn't work. That's my favorite, pictures of chickens with teeth. I love that. One last thing, though, on this idea of comparative anatomy and comparative development, and here's the one that's near and dear to my heart. So I like to study signaling pathways inside cells. How do they get information from outside, transmit it into the cell so it can do something? Maybe it needs to divide, maybe it needs to migrate, maybe it needs to make some new signaling proteins, maybe it needs to turn into a bone, maybe it needs to die. How do those signaling systems work? We're really interested in them. We tend to focus on what's going on inside the cell. A lot of the signals come from outside the cell, and there are just a few signaling systems that are used over and over again by all of the animals on the planet. 7 major signaling systems. Here's one example of those 7. It's called Notch and Delta. Whenever you hear weird names like that for a protein, you can blame the fly people. They always come up with the most bizarre names for their mutants and their proteins. The next thing you know, you sound like you're drunk when you're talking about their work. All right, so Notch and Delta doesn't sound that bad, but if I say ether-a-go-go, then you know something's wrong. That's the name of a protein in Drosophila. Thank you, fly geneticists. We all sound like morons now. Anyway, Notch and Delta. How does it work? Well, one cell has a notch protein on it, just a protein sticking out of the surface of the cell. The recipient cell has the receptor, which is delta. And somehow those, when they touch each other, transmit information into the cell that has delta. What's so interesting about that? Actually, not much. It's the signals that are interesting. The 2 proteins are just kind of sticky and they stick to each other. Except every animal on the planet does this. For some reason, every animal on the planet has decided that when those 2 cells want to exchange that kind of information, which is usually, why don't you turn into a nerve cell now? They always use the same system. There's no good reason for that. It would be easy to design any sort of lock and key mechanism hooked up to the stuff downstream that could send that message. We don't have any other explanation for notch and delta in every animal on the planet except they came from a common ancestor that used it and nobody's bothered to change it. Jot down WNTs, WNTs, if you want other examples of that to go Google in your free time later after you study for whoever's physics test. There's no physics test tomorrow, is there, Lauren? He has to think. That's bad. All right. That phenomenon I just described to you, by the way, signaling systems that are extremely well conserved all through the animal kingdom. It's called deep homology. It's a great phrase. And it's very common. All right, that brings us to, the kind of more challenging stuff. This is a lot of the newer data. This is the kind of data that's being acquired as we speak, being dumped into gigantic databases as we speak, often yet unmined for conclusions. It all started when some crazy loon back in the 90s said, hey, why don't we try to sequence the entire human genome? By the way, that person was almost shouted down. It wasn't one person, but there were very smart people who said, don't be ridiculous. And as you know, it was finished early, and now you can sequence a human genome for, I don't think it's down to $1,000 yet, but it's getting pretty close. Evidence from genetics, loosely defined, that's explained by common ancestry. So first of all, do you know what chromosomes are? You might not. Let me explain to you what a genome is, ever so, there's that word again, not vaguely, ever so simply, using a metaphor that Francis Collins uses that works really well. You got this DNA in your cells that's got all the instructions for doing everything, including making you in the beginning. That DNA is, think of it as a set of encyclopedias, a really big set of encyclopedias. But it's a set so that it's divided into volumes, in this case 23 volumes. You got 23 volumes of that encyclopedia from your mom, 23 from your dad, total of 46. So within those volumes, in each individual volume are little chunks of information we might call genes. Think of them as chapters. All sorts of stuff between the genes that we're still trying to figure out what it does. So it's a bit of an interesting, kind of messy, as yet hard to understand encyclopedia, but that's what it is. But the volumes are called chromosomes. And it turns out, interestingly, that when you look at chromosomes and compare them between, say, rats and humans, let's go with mice actually, mice and humans, you see something kind of weird. On a human chromosome, and they're numbered and they're mapped, and we know what each one is like, there are huge chunks of genes all in a row, and when they're all grouped together, we call that synteny, and so all the genes on chromosome 21, for example, in the human, are in synteny. Sorry, all the genes on chromosome 21 are in synteny. Big chunks of just, think of them as chapters all in a row. Turns out, if you look at the mouse genome, you'll see humongous chunks of what looks like the human genome, everything in the same row, on one chromosome. In other words, the structure of the chromosome is almost the same as the human in a mouse. Now, they're not identical, but they're close enough that someone once thought that they might make a mouse model of Down syndrome by simply making a mouse that had 3 copies of chromosome 16. Biologists in the crowd will know Down syndrome is caused by an extra copy of human chromosome 21. The reason these scientists thought maybe an extra chromosome 16 in a mouse would cause Down syndrome is because chromosome 16 in the mouse looks just about like chromosome 21 in a human. It's not completely the same. It's not like 60%. So start there. What is it about this encyclopedia that everything's kept in the same order in organisms that are not the same? I kind of need an explanation for that. And I can think of 2 possibilities. One is common ancestry. They're copied from the same place. The second possibility is that there's something about that order that matters. It's got to be in that order and it won't work. We have no evidence that that's the case. In fact, people can get along just fine if you take pieces of their chromosomes, turn them backwards. It's not true that we know that, no, dude, it's got to be this one and then that one and then that one. You don't know what's going to happen. Synteny, chromosome fusion. Here's a story that Francis Collins has made famous. You can take, now this is a little risky, but you can look and compare the genomes of 2 very well-known species on this planet, Homo sapiens and the chimpanzee. I don't know why you would do that, but you can compare those 2. And you can make maps of all the chromosomes, and you'll see something pretty extraordinary. They look bloody well the same. They look like they're the same stinking genome. Well, not really. There's a couple of differences. I'm not talking about comparing little letter by letter. I'm talking about the structures of the chromosomes. There's one interesting difference. It turns out chimps have more chromosomes than humans do. They have one extra pair. So here's a picture of what it looks like when you build a map of a chromosome. And folks, this is not the same as sequencing the DNA, different thing. Here's a map of the chromosome, and these little bars indicate some staining technique that shows that this is a particular region of this chromosome and that's one. And you'll notice that these two chromosomes here, they line up really well, right? These patterns are almost identical all the way. This is the chimpanzee chromosome. There's one, and there's another one. And here is human chromosome 2. Human chromosome 2 is evidently a fusion of two chimpanzee chromosomes, linked tail to tail. If you put that onto that, you get this. In fact, what's even more interesting is these sequences that are here at the end of this chromosome, there's a particular kind of DNA that's at the end of chromosomes that are called telomeres. We know what telomeres look like, and so if we're right that this thing is a fusion of two chromosomes from a chimp-human common ancestor, there should be telomere-like stuff right in the middle of a chromosome. Interestingly, there's telomere-like stuff right in the middle of that chromosome. I see why this is Francis Collins' sort of favorite example. Common ancestry makes a simple explanation here. So we've got a common ancestor that had two chromosomes. Ours has since fused. Theirs remains separate. Design arguments, you know, I haven't heard any good ones on this count. Sorry, I just gave away a secret. Genetic fossils. Actually, no, I didn't. So this might interest you. You know, no offense, you're terrible at smelling. This is why we have dogs, right? Have you ever heard of a bomb-sniffing toddler? No. Bomb-sniffing dogs, because they're really good at smelling. We stink at smelling. It's a joke. I think you might be the first one that laughed at that, Lauren. That's not good at all. All right, so actually that was not planned. And I'm like, why are people laughing? You know, note to self, stink at smelling. We're no good at smelling compared to other species. Why? Well, lots of reasons, but here's one. We have receptors in the olfactory neurons up here in the nose. The olfactory neurons are cells that have a receptor on them that will recognize some kind of thing in the environment, an odorant, and send a signal into the brain. And then all the different sort of things that bind in your nose all get added up in your brain to brownies. All right, so that's roughly how it works. We've got these receptors. There are 800 olfactory receptor genes in the human genome. More than half of them are dead. They've been mutated in a way that looks for all the world to us like they can't make a functional protein. And yes, we can show that they don't make a functional protein. Mice have 1,400 of these receptors. About 25% of theirs look like they've been mutated so that they don't function anymore. Mice have more receptors than we do. Mice smell better than we do. Well, you know what I mean. They have more olfactory acuity than we do, if you need the technical jargon. Our genome is littered with these things. We call them pseudogenes. We know that they've been mutated in such a way that they can't make a functional protein. And yes, we can test to see whether that's the case. Most famously, we all have to take vitamin C or consume